Hello everyone, I will be continuing Nathan and Arija's presentation, who have already covered biomaterials and neuroprosthetics, and I'm going to be covering another application of stem cell biomaterials in neurodegenerative disease modeling. And there's a lot I want to talk about more in detail, but for the sake of time, we're just going to cover an introduction to these topics and focus on Alzheimer's disease. So here's the outline. We're going to first introduce the ethics, the discovery of iPSCs, what are iPSCs, regulations surrounding them, disease modeling, materials and methods, and the limitations. And when introducing a biomaterials topic, it's kind of unusual to start with ethics, but I feel that when discussing stem cells, especially if you were cognizant of the politics in the early 2000s, this seemed to me like a natural place to start. So there's this property called pluripotency that we're ultimately after. And we'll talk more about that later, but before 2006, the only way you could get pluripotent stem cells was from embryos because their cells are yet to be differentiated further. And I'm going to highlight that the discussion of ethics surrounding stem cells mainly involves the donation of biological materials, stem cell clinical trials for potential therapies, and the destruction and creation of embryos for stem cell research. And as you can imagine, there could be many arguments against this destruction and creation, and it begs the question, is the embryo considered human life? And this used to be a huge ethical barrier for scientists at the time. So if we could somehow bypass the need to use embryos, that would be very useful to us. So useful, in fact, that this discovery won the Nobel Prize in 2012 for the discovery that mature cells can be reprogrammed to become pluripotent. And this was discovered by Yamanaka and Gurdon. And according to Yamanaka, this discovery was actually driven by him seeing an embryo for the first time and likening it to his daughter. So you may still be wondering, what's the big deal with pluripotent stem cells? Now, pluripotent stem cells are undifferentiated cells with self-renewal capability. And these cells have the potential to differentiate into any cell type of the body. Now, induced pluripotent stem cells, or iPSCs, are somatic cells that are genetically reprogrammed, essentially backwards to an embryonic stem cell-like state, meaning embryonic stem cells are no longer our only option to obtain pluripotent cells. And after this discovery, the field really took off. And the global stem cell market is now expected to reach $15.63 billion by 2025. And research is continuously expanding the possibilities as new applications are being explored, especially to treat diseases. And some of the sectors include manufacturing cells, producing biologics, compounds and materials, precision and regenerative medicine, and research to better understand human function and diseases. And even though we no longer need embryos to obtain pluripotent stem cells, with all the research and promise surrounding stem cells, I also wanted to briefly highlight that an important aspect of regulations today is still consent. And an individual needs to consent to their cells being used for research. So how are iPSCs useful to us today? Well, in the limited time that I have, I want to discuss their use in modeling because right now we really need better models with which to model disease. And today there's yet to be a drug or cure for Alzheimer's, and it's fair to suppose that it could be that what works for rodents simply doesn't work for humans. And in this domain, they like to say, it's a good time to be a mouse with Alzheimer's. But none of these findings have been successfully translated yet. And rodents can simply never fully model human pathology, and it's usually not ethical to use humans for laboratory experimentation. And to illustrate this need for better human models, here's an example of a tragic clinical trial where they had a fatality and several hospitalizations. And this particular drug was totally fine in rodents and unexpectedly produced these effects in humans. And if you're interested, you can read more about it at this link here. But with the advent of iPSC technology, we can now study human neurodegeneration by culturing patient-specific iPSCs taken from a patient's somatic cells. And these cells are shown to preserve many of the pathological signatures. So we don't need a priori knowledge of the genetic cause of a disease, and we can further perform gene editing on these cells to create isogenic lines if desired. And Silva et al. Reviewed the validity of these iPSC models taken from several papers and found that patient-derived neurons were showing pathologies that were validated in human brains post-mortem, like amyloid beta accumulation, ER, endosome, and lysosome toxicity, and oxidative stress. And 3D organoids showed A-beta aggregates and tau tangles as well as endosome abnormalities. And here's just a brief schematic on how drug discovery might work, according to Penny et al. From a lab doing this kind of work at MIT, where they take samples from AD patients and control healthy individuals, reprogram them back to iPSCs, and then culture neurons, astrocytes, microglia, oligodendrocytes, parasites, and vascular endothelial cells to generate a 3D in vitro brain model, complete with a functional blood-brain barrier. And they can then use compound libraries to identify new drugs in a high-throughput manner. And Ochalek et al. 2017 showed that in Nestin and PAX6 expressing neural progenitor cells derived from iPSCs from both sporadic and familial AD, they show characteristic elevated expression profiles of A-beta when compared to healthy control. So briefly, this slide, as briefly as possible, goes over the steps for generating neurons from iPSCs. But of course, you should refer to a full protocol. So first, you generate stem cells by adding retinoic acid or inhibiting BMP and TGF-beta pathways. And then you can add growth factors to direct differentiation to specific neuronal subtypes. To model AD, you can generate cholinergic neurons by targeting the REST and co-REST signaling pathways. And then using FAC sorting, you can select neurons by gating for specific markers, since the population is probably going to have some heterogeneity. And again, a very brief word on materials used in stem cell research. Whether stemness is maintained or differentiation takes place is dictated by what they call the stem cell's niche in our bodies, which is basically its environment, including surrounding tissues, cells, and other factors. So in order for a stem cell to behave a particular way, you may need materials of a certain stiffness for them to grow on, or need to be co-cultured with their other cells, depending on what they're used to, so to speak, in their niche. So they can be grown on collagen or laminin, or on or within hydrogels, for instance. And by far the most common container for them are your typical polystyrene plastic tissue culture plates. And as you know, polystyrene is ideal because it's transparent, it's easy to sterilize, but they're also hydrophobic, so they typically need to be coated to promote cell adhesion. And this slide just covers some of the limitations in these iPSC-AD models. And that includes reliability. For instance, you have the possibility of variability between clones. Like we mentioned before, some protocols generate heterogeneous populations of the target cell type. So you would probably need to isolate your cell type of interest with FAC sorting. You have the absence of vascularization and microglia. However, we had an example before where different labs are actually tackling this problem by using 3D models that involve culturing BVECs and microglia as well. And some models have limited imaging capabilities and can be quite costly. And there's also a question of whether these models are age equivalent, because the primary risk factor in Alzheimer's disease is age. So just to summarize, induced pluripotent stem cells are somatic cells that have been genetically programmed to an embryonic stem cell-like state. Rodent models don't fully recapitulate human pathologies associated with age, genetics, and environment. iPSCs can be used to generate human-derived models of neurodegenerative disease, but this field still has its limitations and further advances are needed. And here are the references. I think we're just about a little bit over 10 minutes. That concludes my presentation. Please feel free to reach out if you have any questions, and thanks so much for your attention.